Hey everyone, welcome back to studying about molecular inheritance. We have previously gone over DNA, the structure of DNA, the scientist who discovered the structure of DNA, and Erwin Chargaff, who actually told us the rule of A equals T, C equals G. But now we're actually going to move into RNA. So we're finished with the topic of DNA, but that doesn't mean we can forget anything that we learned about DNA. We are just moving into RNA, which is even more advanced than DNA. So RNA stands for ribonucleic acid, and it's made up of nucleotides containing the sugar ribose. So remember that a nucleotide is made up of your nitrogenous base, your phosphate group, and a sugar. The sugar here is going to be ribose instead of deoxyribose, which is the sugar that made up a nucleotide for DNA. So we've also got those four nucleotides that make up an RNA molecule. And the reason there's four is because remember we're going to have different nitrogenous bases. So we can have adenine, uracil, cytosine, and guanine for those nitrogenous bases for an RNA molecule. Now you might be thinking, what happened to thymine? Well, uracil actually replaces thymine whenever we are looking at RNA. RNA is a single-stranded, okay? So it's not double-stranded like DNA was. It's actually single-stranded. But sometimes it will wrap around itself, which can allow for base pairing. But typically that's not going to happen. RNA is a helper to DNA. What this means is that it allows protein synthesis to occur according to the genetic information that was stored in DNA. And there are three types of RNA. So we can have messenger RNA, transfer RNA, or ribosomal RNA. So the first is that messenger RNA. This is produced in the nucleus of a eukaryotic cell and the nucleoid region of a prokaryotic cell. So mRNA is formed through a process called transcription, in which DNA genes are transcribed into mRNA. So mRNA comes directly from DNA. The mRNA carries genetic information from that DNA molecule in the nucleus to the ribosomes in the cytoplasm, where protein synthesis occurs through a process called translation. So you have to have that mRNA occur because mRNA is what is grabbing that information from the DNA, taking it outside of the nucleus to the cytoplasm to that ribosome. We then have messenger RNA. This is also produced in the cell nucleus of eukaryotes. tRNA transfers amino acids present in the cytoplasm to the ribosome. There's also 20 different amino acids, and each has its own tRNA molecule. At the ribosome, a process called translation occurs to join the amino acids to form a polypeptide chain. So this tRNA molecule is going to have that anticodon right here, and each is carrying an amino acid. And these codons will form together on the ribosome. Then ribosomal RNA. rRNA is produced in the nucleolus of the nucleus, where a portion of DNA serves as a template for its formation. rRNA joins with proteins made in the cytoplasm to form subunits of a ribosome. These subunits leave the nucleus and come together in the cytoplasm when protein synthesis is about to begin. So, the three types of rRNA. You've got your messenger RNA, you've got your ribosomal RNA, and you've got that transfer RNA. This really moves us into this central dogma of molecular biology. This states that genetic information flows from DNA to RNA to the protein. So you've got DNA, which has your double helix. So it's going to be transcribed into RNA, which is a single strand, which will then be translated into a protein. Now, what does all this mean? Well, with transcription, this takes place in the nucleus. During transcription, a portion of DNA serves as a template for your mRNA formation. Translation takes place in the cytoplasm. During translation, the sequence of mRNA bases determines the sequence of amino acids in a polypeptide. So, molecular genetics. How do we fit all of this together? We've learned about DNA, we've learned about RNA. Well, molecular genetics tells us that genetic information lies in the sequence of the bases in DNA, which through mRNA determines the amino acids in a protein. tRNA assists mRNA during protein synthesis by bringing amino acids to the ribosomes. Proteins differ from one another by the sequence of their amino acids, and proteins determine the structure and the function of cells. So your genetic code. The information contained in DNA and RNA are written in a chemical language. This is a triplet code, which is the 4 bases of RNA can supply 64 different combinations for this triplet code. Each 3-letter unit of an mRNA molecule is called a codon, and it codes for a single amino acid. There are 61 codons that correspond to a particular amino acid. The remaining 3 are stop. The amino acids may have more than one codon, which offers some protection against possibly harmful mutations that can occur if there's a sequence of the amino acids that is messed up in that protein. And so, what this means, if that first letter of the codon is a C, then I would go right here. So the first letter of our codon might be a C. So I would look in this row, okay? Now, if the second letter is an A, that means I am going to be in this box. So C and A. And then I look on this row, and if it's a U, so CAU, then it's going to be CAU, and that codon represents the amino acid of HIS. Okay? So, use that book, use your textbook as a reference. This is really just a big overview of what this chapter has to offer. Make sure that you are looking at your resources that are available for you, as well as your textbook and all of those resources. They will help you get through this. Please let me know if you have any questions regarding any of this over RNA, so mRNA, tRNA, or rRNA.